Hello, Science 30. We are going to talk today about genetic technologies. Essentially, genetic technologies are just different things that help us modify our genetics, whether it's gene therapy or cloning or something along those lines. All right, we're going to talk about different technologies that allow for this to happen. So before we do, we're going to watch this quick video to have an idea about what is genetic
So,
engineering.
what is genetic engineering? Genetic engineering is when the genetic code of an organism is altered to produce a desired trait. This is done by taking a gene from one and inserting it into the DNA of another to get the wanted end result. To get a better sense of how this is done, let's first understand how DNA works. In any living thing, the DNA acts as the instruction manual for the body. Certain genes code for certain proteins, which in turn make up all the different parts of the organism. Any trait that you can think of is coded by DNA. Now, once you know what codes for what, you can use genetic engineering to essentially edit the parts you want, to some extent. All living things use the same genetic code, DNA, made from the same 4 bases, so scientists can use special enzymes to cut parts of the DNA from one living thing and sort of sew them onto the DNA of the target. The result is then a living thing that has a new gene and a new feature as a result of that gene. Now, there are many examples where genetic engineering is used today, but we're going to focus on a very specific medical example to understand this concept better. Humulin, a medication used for diabetics, is created with genetically modified bacteria. Humulin can be given to diabetic patients who lack the insulin they need. Now, how they do this is scientists figured out which section of our DNA codes for insulin, and they also found certain enzymes that can cut that gene out. What they do is, using enzymes and heat, they isolate specific gene and then insert it into the DNA of bacteria. The result is now a bacteria that produces human insulin. And since bacteria can multiply really fast, you can create a lot of insulin in a very short amount of time. Now, this is great. Genetic engineering poses huge potential in the field of science, providing us with new insight on how our genes work. However, there's also a lot of controversy in certain respects to genetic modification, specifically genetically modifying our food. To learn more about GMO foods, check out our next video.
So hopefully from that little bit of a video, you started to understand what is genetic technologies and genetic engineering. So genetic engineering is used to cut DNA from one organism and paste it into another. We call this process splicing. Essentially, over the years, we have looked at different ways that animals breed in humans, and we've said there are ways that we can do this better in some aspects. If you think about dogs, for example, purebred dogs. If you want a purebred dog, you need to breed one dog and another that came from the same line, okay? And they are 100% that version of that dog. The problem with doing purebreds is that a lot of times you get genetic diseases that get passed along, bad hips, bad eyes, and stuff like that. So actually having a dog with a variety of mixes is better. And what we are seeing in regular practices of breeding is that sometimes when you breed 2 animals together, you end up with a better animal in the end. So they took this idea, this refinement of traditional breeding practices, and said, you know what, if we breed one thing with another, we can make something better. Could we just do that right away at the genetic level? So we ended up eventually from mitosis, remember that idea of making clones, looking at technology that eventually allowed us to do things like cloning experiments. Now, when we talk about cloning or any sort of genetic engineering, but specifically cloning, we're going to look at a couple different things. Okay, these are the 4 things we're going to need. Essentially, the first thing you're going to need is a plasmid from a harmless bacteria. So E. coli, you guys might be like, oh my gosh, that's not harmless. That's really dangerous. You can get really sick. Truth of the matter is that you have E. coli in your guts, and they're actually really, really good for you. It's when you eat meat that is tainted with E. coli that it's bad. So what they do is they take a plasmid, which is a circle part inside the bacteria. So if you're looking at the bacterium picture, there's a plasmid of a yellow round circle. Plasmids are very cool because they actually produce separately from the DNA of the bacteria. So they do their own thing. So what scientists do is they take it out and then they find the gene they'd like to clone. So in the case of the insulin, they found the human insulin gene. They then use something called a restriction enzyme. And a restriction enzyme is what we use to cut bacteria or DNA. So they use a restriction enzyme, cut out the actual gene, and cut part of the plasmid. And then they use glue, or what we call ligase, essentially, and then they put it all together. So those are the 4 things that we need to do to start this idea of creating new insulin inside different bacteria. So why do we use E. coli? Well, it's kind of really interesting. E. coli, like I said, isn't harmful to you. You have it in your guts, but it's super easy to make because there's lots and lots of it. It's cheap and it's really easy to manipulate. It also has a really short lifespan, so meaning that you don't have to wait a really long time for it to replicate. So they have small circular rings of DNA, like I said earlier, that are called plasmids, which we use to insert our genes into that we want to replicate. Plasmids replicate separately from the DNA of the bacteria. Okay, so you can see the red DNA in the bacteria there, and then the blue plasmids. They actually don't replicate together. So we're not actually doing anything to the bacterial DNA. We're using something that replicates separately. Plasmid replication, like I said, does not affect anything with the bacteria's genome. It does it all on its own. All right, so in the picture, you can see isolated plasmids, ones that we've taken out of the bacteria to use to put in an isolated gene. Now we're going to talk about what is a restriction enzyme. Now, a restriction enzyme is used to cut DNA at specific recognition sequences. So that's a really important sequence. They're usually from about 4 to 8 nucleotides, so A's, T's, C's, and G's, and they're usually what we call palindromes, meaning if you look at them one way, so T, C, G, A, if you go backwards up, it's T, C, G, A. So they run opposite of each other, but they're palindromes because if you go down, it's the same, and if you go up, it's the same. Now, restriction enzymes, there's about 3,000 different ones. We have 600 of them commercially used and available that can cut DNA. And one that we use specifically comes from a bacteria called E. coli, and this is from E. coli. It uses its restriction enzyme as a defense mechanism against bacteria. So the bacteria against viruses. So what the bacteria actually does is when a virus tries to invade it, it can actually cut and snip the virus's DNA or cut and snip the virus so the virus can't replicate. Now, what's really, really interesting about this is this idea of bacteria being able to have this defense, this restriction enzyme against viruses, is what created something called CRISPR. And CRISPR is a newish, probably has been around since 2017, restriction, in quotations, restriction enzyme. It's actually a very cool little set of DNA sequences that can go in and snip DNA. And we're going to talk more about CRISPR later. It's very interesting, but it comes from this idea that bacteria already naturally had this defense to back off, to get viruses to back off. We then have now used it and taken it even further to say, hey, is there ways that we can use this to help solve things like diabetes or HIV? So what it does, like I said, is it selectively cuts foreign DNA. So when the virus attaches itself to the bacteria, it can go in and cut the virus's DNA so that it can't replicate. So what we're going to talk about is the steps and processes that we are going to go through in order to do this whole idea of genetic engineering of a protein or of a insulin hormone. So the first thing you want to do is you want to take that plasmid from the E. coli bacterium and cut it using restriction enzymes. So again, we're just cutting the plasmid. We're going to take the DNA, the desired gene, from the chromosome and use the same restriction enzyme. So we're looking for insulin. We're going to go in. We're going to cut out that insulin gene. We're going to place both the plasmid and the isolated gene in a test tube, and then we're going to glue them together. We're going to remove the recombined plasmid, so that just means that it has new genetics to it because now it has a new gene added to it. And we're going to put it back into the E. coli bacteria and place the bacteria into a petri dish. Okay. As a reminder, because the plasmid will make this gene separately than its DNA, when the bacteria grows and then splits through the process of binary fission or whatever, it mass produces the protein that's inside that desired gene because those plasmids are just replicating all on its own. Then what we do is we go to that plasmid and we go and we take that protein, we isolate it, we sterilize, we bottle it, and then we sell it to the pharmaceutical industry. So those are the steps that it takes in order to create something like insulin. If we're looking at it in a process in a picture, you can kind of see it here. On the right side, we have bacterium, and on the left side, we have the human cell. So we go in, we slice up the chromosome, we take the little piece that we want for insulin. On the other side, we take out the plasmid and we cut the plasmid. Then we put the two together. We replace the plasmid back inside the bacterium, and then we let the bacterium reproduce. And eventually, you're going to get a lot of different types of plasmids with the protein on it, and we can then take it and produce it and bottle it and sell it. All right, so this is a longer video on CRISPR, and I'm going to post it separately. We're not going to watch it in this video. I'm going to post it separately. All right, so we're going to talk about gene therapy. Gene therapy is essentially following the success of genetic engineering. Scientists pose the questions, could mammalian chromosomes be modified to treat genetic diseases? Now, gene therapy is still largely in its clinical stages. When you think about diseases that only directly affect you, so you're an adult and you have diabetes, okay, or you have some sort of cancer, is there a way to use gene therapy to help treat any of these diseases? Now, what it is it's a gene replacement is administered to a patient with a defective gene using a viral vector. They're trying to treat things like sickle cell anemia, diabetes, cystic fibrosis. All of these have been looked at. So what they're essentially trying to do is they are trying to take disorders, put in a gene that will work, and try to turn off the gene that is. We're going to just kind of take a quick look at what this looks like. So what happens is cells are removed from the patients, and the laboratory, a virus is altered so it can no longer reproduce. Okay, so we don't want the virus to reproduce. We just want it to be able to have the gene that is good inserted into it. So we take the altered virus and we mix it with the cells of the patient, and then the cells from the patient becomes altered, and then that altered cell is put back into the patient, and then we hope that it produces cells that actually have the desired protein or hormone. So essentially, it's taking cells that are not working, creating cells that are working, and hoping those cells divide. So here's another quick video that we'll watch just to give you an idea about what is gene therapy.
So what exactly is gene therapy? First, let's start with the basics. This is a gene. Genes are made up of DNA, which are the blueprints to build the enzymes or proteins that do the work for your body. As far as we know, humans have between 20,000 and 25,000 genes. And most people have 2 copies of each gene, inherited from each of our parents. They are the building blocks of our physical makeup, from the color of our hair, to our athletic abilities, to our immune system. But sometimes genes aren't built correctly. And a small adjustment to the DNA within our genes can change how our proteins work, which can affect how we breathe, walk, or digest food properly. To better understand how genes change, here's Dr. Michelle Kalos.
So our genes are mutating all the time as a result of chemical processes in the cell, as well as external influences like ultraviolet light. We can also inherit mutations from our parents. Now, when we wind up with these mutations, they can sometimes cause disease, and that's really where gene therapy comes in.
If genes are like the blueprint to our body, gene therapy can fill in some missing drawings. Gene therapy is the introduction, removal, or change in genetic material, specifically DNA or its molecule cousin, RNA, into the cells of a patient to repair or change a gene to treat a specific disease. The transferred genetic material then changes how a protein, or group of proteins, is produced by the cell. It can reduce levels of certain disease-causing proteins, increase production of disease-fighting proteins, or even produce new or modified proteins within the cell. You may be curious how a gene and the genetic material it holds is transferred to the cell during gene therapy. Well, a gene cannot be directly inserted into a cell. Instead, a carrier, also known as a vector, helps deliver the gene. Typically, viruses are used as vectors because they've evolved to be very good at sneaking into and infecting cells. But in this case, their motive is to insert the gene or genes into the cell. The infectious part of a virus is removed, allowing it to still enter the cell's nucleus without making the patient sick. It's worth noting there are two kinds of gene therapy, in vivo and ex vivo.
In vivo gene therapy, the genes are introduced directly into the patient's cells while the cells are still in the patient's body. In ex vivo gene therapy, cells are removed from the patient's body, then the genes are added to those cells in the lab, and the cells are then returned to the patient. The right approach depends on the disease that the patient has and the tissue that's affected, and that would tell us which is the best way to approach this.
Advances in gene therapy research allow us to treat and hopefully someday avoid rare and debilitating diseases. For more information and resources about gene therapy and how it works, go to ASG.
So that's a little bit more about gene therapy and the basics. We are going to get into depth a little bit more, like I said, with CRISPR and discuss that, as well as we're going to watch a little video about designer babies later. So if you have any questions or concerns, please let me know.